The things I'm going to cover today, because I see the questions that are coming up, is complete dominance, which we've done now. Incomplete dominance and codominance and the difference between the two. I'm going to be going through Mendel's laws. There are 2 of them that you have to know. All right? And then I'm going to go through mutations and what the different mutations are. So what a frame shift mutation is and what a point mutation is. And once I've done this with you, you are going to be A for away. Okay, so just be patient. Just let me get through it. All right, so here we go. Mendel's law, and I actually asked John, because, you know, I normally write everything myself. In fact, let's go with nice yellow. It's nice and bright and breezy. It's his law of dominance and segregation. Now, in some books and in some countries, they talk about Mendel's law of dominance. In others, they talk about Mendel's law of segregation. So I decided, in case there's any confusion, it is Mendel's law of dominance, which is segregation. And what he said was that when there are 2 individuals with contrasting, means opposite, homozygous, which means purebred, you know that, characteristics. In other words, we're going to have an example that we've just done, a capital B and a capital B. Both the alleles are dominant, but they're homozygous, they're pure, times little b, which would be the white, so brown versus white, 2 contrasting homozygous, which means they are pure, characteristics, are crossed. The individuals of the F1, that's the first family generation, hybrid, because they're a mixture of the 2 parents, will all resemble the parent possessing the dominant characteristic. Because it is, people look here, the law of dominance and segregation. So what it's saying is, if I've got a homozygous parent that has the 2 dominant, because remember it's homozygous, so both alleles are dominant, and both alleles are recessive for the other parent, because they're homozygous. And I cross them. This was our first cross that we did. If we take capital B, what am I got? I've got capital and small, and capital and small, and capital and small, and capital and small. So I'm going to end up with 100% of the offspring are going to resemble the parent with the dominant gene. Okay, but they're all carrying the recessive, they're carrying a recessive gene there, but they are going to resemble the parent with the dominant allele pair, the homozygous dominant parent. That's Mendel's law of dominance and segregation. It's easy. 2 individuals with contrasting homozygous, which means pure, I just put it in brackets there. That's for you. Homozygous genes, all right? When they are crossed, the F1 generation, which means the first family, the F1 generation will all resemble the dominant parent, even though they are heterozygous. Okay, very important. Then we go all the way up to the top. That was the slide I missed. Okay, so what I did here was I did this for you. And I said, right, in the first parent generation, what do I have? I've got, now this is what Mendel did. He worked with sweet peas. He worked with peas, flowering peas. Okay, and he said, okay, well, you remember he was a monk. He made this little garden and he played in his garden. Okay, so in the first parent generation, I have the tall parent and the short parent. So homozygous dominant, homozygous recessive. Okay, the first lot of babas that come, the first group of little seeds, when you plant them and they grow, what do we see? We see tall plants. Why? Because their characteristic, this dominant characteristic, is now seen in the phenotype in each of the babies. All right, each of the seeds in this case, because they're plants. All right, now I take those seeds, all right, and they become the second parent generation. So they're the second lot of parents. And we're going to cross these plants and we're going to say, right, what have we got? They are hybrids. In other words, a mix of the mom and the dad, the male and the female, the whatever, they are a mixture. And being hybrids, they are capital T, small t, which means they are heterozygous. In other words, they have one of each gene. Now, what have we got? Tall times tall is what we see in the phenotype, but in the genotype, what is in the genes, capital T, small t. So wow, look at this. Capital T in the F2 generations, the second lot of babies. And then we have 50% or 2 out of 4. This is 1 out of 4. 2 out of 4 are heterozygous. And guess what? 1 in 4 or 25% will be short. There's my recessive gene. Why? Because one little t came from mommy and one little t came from daddy, or one little t came from the egg and one little t came from the pollen grain or the sperm, in this case the pollen grain because we're talking about plants. Okay, and this was, remember combination 3 that I showed you? Here it is. And this is probably the most popular one. And what it does is they tell you, explain Mendel's law. Easy. Show us. Well, piece of cake. Homozygous parents, dominant, recessive, cross, heterozygous babies. All of them show in the phenotype the dominant gene. Cross them. Hello, baby. How do we know that they're heterozygous? Well, look at the baby there. All right. This offspring here, homozygous recessive. It's a 1 to 2 to 1 ratio. And in the phenotype, 3 of them are going to be tall and only 1 is going to be short in the phenotype. And remember, we're talking about percentages here. Okay? Not actual numbers. All right. This I found on the internet. It was so pretty, I couldn't resist it. Okay. This is his law of segregation or dominance, as we said. Here's the first parent generation. We've got pure red sweet peas, pure white sweet peas. So if capital R is red, small r is white, and guess what we're going to have? So this is applying Mendel's law. Mendel's law, or Mendel worked with tall and short plants, and then he went colors. So here we go. What have we got? We've got the red gametes separate during meiosis. The white gametes separate during meiosis. What are the combinations? I have all 4. So 4 out of 4 are going to be the hybrid red. That's your first generation. It is your F1 generation. Now we go on to Mendel's first law of segregation, but what do we have here? We have our hybrid babies that are now parents, so it's P2. Okay, and what do we have? We have during meiosis, the gametes separate out. So capital R, small r, dominant, recessive, and look at what we end up with. We end up with 1, 2, 1 in 4, and there's my recessive. Okay, this is for complete dominance. Okay, now we move on. And okay, I put this on because I thought it would be interesting for you. Dominant and recessive. Unless they tell you in an exam, it is the other way around. They could say the mother is blonde with blue eyes, and she dominates over the father with black hair and brown eyes. All right. Unless they specifically tell you this is dominant and recessive here. So brown will dominate over blue. Curly will dominate over straight. And by the way, just as a snippet here, eye color, hair color, those are, you have multiple alleles. So we call it polygenetics. Okay, why? Because there are many different genes that work on one characteristic like eye color, hair color. So we do very simplified versions of genetics in your life science. Even though it's difficult for you, we're giving you really the basics. We're giving you the 2 times, the 5 times, the 10 times, and the 11 times tables out of all those numbers. You follow? This is really just the basics. So straight and curly hair, black and white, normal skin color, by the way, eye color, skin color, height, those are all polygenic. So you need lots of different genes, allele gene sets that determine it. All right, and then tongue rolling. Indy, can you do this?
I actually can.
You see, why? We both can, 2 out of 2, because it is a dominant gene. All right, so what have we got here? Got dominant brown eyes versus blue, curly hair versus straight, black hair versus white or blonde, normal skin pigment versus albinism. Okay. Tongue rolling versus a flat tongue, which we've just gone through now. Looped earlobes versus straight. Now, I want you to feel your ears, or if you've got something that reflects or a mirror, have a look. And you'll see some people have a looped earlobe. So it does, if this is the face, the earlobe comes, the earlobe will do this. It's got like an indentation there, and I've just moved this. Come. If this is the face, you've got the ear does, oh no man, the ear does that. Whereas a straight one, if this is the face, or let's go the face that way, the ear comes and it's straight like that. All right, so whether it goes in or whether it's straight, normal blood clotting versus hemophilia, but hemophilia is recessive, but it's carried on the XX chromosomes. So in other words, it is sex-linked. And then you've got your blood groups. And if you look at the blood groups, people, you've got A, you've got B, are dominant, and you've got O is recessive. Right, so this means it is polygenic as well. You have multiple alleles, more than 2 alleles for a characteristic, because how many are there? There's 1, 2, 3 for blood groups. And this is where we're going to explain codominance. We'll get there though. All right, here we've got incomplete dominance. And this was the best way I could explain it, is if I have P1, which is the first parental process, we have homozygous dominant, okay? We have homozygous recessive. They are homozygous, they are pure, dominant and recessive, and our first filial generation, our first little set of babies, what are we going to have? We are going to have all of them pink. So if I take red and white and I mix it like paint, I have my hybrids, which are the babies, and the hybrids are a mix of the dominant and the recessive, because the dominant doesn't dominate completely over the recessive gene. So you have a mixture. So let's take this. If I have black and white, I'll end up with, come, think, gray. Perfect. You follow? If I have curly hair, which is dominant, and straight hair, which is recessive, and I mix the two, what am I going to have with incomplete dominance? I'm going to have wavy hair. It's not curly, it's not straight, it's in between. You know, it's like Little Red Riding Hood. This porridge is Goldilocks and the Three Bears. This porridge, Daddy's porridge is too hot, Mommy's porridge is too cold, Baby Bear's porridge is just right. So red, white gives you pink. Black, white gives you gray. That is if it is incomplete. In other words, the dominant gene does not dominate completely over the recessive gene. And they sort of strike a bargain and a compromise. It is a mixture of the two. And get that in your head. It is incomplete. It can't dominate completely. Okay? Now, codominance. And people, the learners mess this up year after year after year, and it's so easy. Complete dominance, doesn't matter what that recessive gene is. Unless there are two recessive genes, there is no way that trait shows in the phenotype. Okay, in the physical, in the phenotype. That's dominant. Complete dominance. Incomplete dominance, it says, you know what? I'm red, you white, let's have a little mixture there. Let's throw a party and be happy, and everybody just compromises. It's a mixture. Codominance is no. You're dominant, I'm dominant. Let's agree that we each have our little patch. And that's what it does. So it says it's means both. Both dominate. Not just one with complete dominance, not incomplete dominance where they say, well, we just wusses. We're just going to strike a compromise, which I suppose is the nice way to do it. It says, we are both equally important. We are codominant. And the reason I used color was because I want to show you a flower. Generally, the examples that you'll get in an exam or in a test will be on blood. But just in case they throw a curveball at you, this is to cover that. Okay? Your P1, first parental process, we have homozygous dominant here. Okay? Capital R, capital R. But here we have white, and the white says, okay, look, I know I'm recessive, but guess what? I'm not actually that recessive. So we have homozygous, but this white also dominates. And I didn't want to use the letters W. So it's also dominant. We have codominance. So what do we end up with here? In the F1, we end up with something that is red and white. Red and white, red and white, red and white. So the red has its place, the white has its place. And you check this out. And this is what we have in nature. Look at this little flower. And if you go to, it's gladewaterbiology.blogspot.com. If you go and have a look at that site, this is a perfect example of codominance. I've got the pink, I've got the white. Okay, it's like taking codominance would be if I have a Friesland cow would be codominance. I have a black cow, a white cow, they mate, and guess what? The babies are black with white spots or white with black spots. Okay, a Dalmatian is a perfect example of codominance. It would be if you crossed a black dog with a white dog, they codominate. We have a Dalmatian, which is, and you all know 101 Dalmatians, so you know what I'm talking about. It's a dog with black spots. Okay, that would be codominance. Now, the example that you need to know for exam purposes is codominance in blood. Now remember, erythrocytes are your red blood cells. Okay, your it wasn't spelled right. Erythrocytes. So your erythrocytes, your red blood cells. Now have a look at this. We have a red blood cell which is antigen A. We have a red blood cell that has antigen B. We have red blood cells that have both antigen A and B. So we call it blood type AB. And then we have a red blood cell that has no antigens. And if it has no antigens, we say that it is O. All right, so O is the recessive one. The only way that trait can come through, come on, if it's recessive, the only way you are going to see that trait in the phenotype is if there are two homozygous recessive traits together, then you will see it in the phenotype. So for someone to have blood that is OO, or what we call O, they must have the two, okay? They must have both. Both alleles must be recessive. A and B say, we are both equally strong, and if I have antigen A, all right, it's blood type A. Antigen B, blood type B. But oh, woe is me, if I have a mother and a father, and the mother is A and the father is B, and those gametes get together, it says, we are working here together, and you end up with AB. Now, look how nice this is. And I put this together for you as well. We've got blood type, what we call, and what the hospital, if you take a blood test and they say, oh, you're blood type A, you B's, A, B, O. This is what it means. Blood type A is written like this. You put a little line with a little dot, and you can't see the yellows, so let's do this. That little thing there is a little line with a little dot, and you put an A. And a little line, and so what it's telling you is that's the chromosome. And on that chromosome pair, you are going to have either A or nothing. In other words, there's no antigen here. And if it has no antigen, okay, that's what you end up with, and that's what you end up with. So they can receive blood. If you A group, you can receive blood from other A's, or you can receive blood from O. That's why we say that anyone with blood type O is a universal donor, okay? Because they can give, a universal donor, because they can give blood to anyone, because O is compatible there, O is compatible here, it's there and here. Now, if it's blood type B, all right, here we've got BB, or we've got BO. All right? So they can receive blood from other B's, or they can receive blood from O's. Then AB. This is where a perfect example of codominance. So to get blood type AB, mom or dad had to be A, and the other one had to be B. And you put the A and the B together, it says, codominance. And that's what we have. So AB, there we go. Now look at this. AB can receive blood from A's. They can receive blood from B's. They can receive blood from AB's and O. So they are what we call the universal receiver. Okay, they can receive blood from anyone. So if you are AB, you're very lucky. It's when you are B, you have a problem, or if you are just A, you have a problem. And O, well, O has no antigens at all. You have the two recessive genes together for the trait to come through. Okay, it's as easy as that.